See, as I told you, we will be discussing all questions related to plant physiology and human biology as such. So I am considering human reproduction also to be a part of human biology. Plant physiology particularly I want to take it up because if we go into plant anatomy, physiology and reproduction, it will get confusing. Human biology, we can consider the entire human biology as a continuum, as a unit. Plants, let us not do that. Easy to understand is physiology because it is related to function. Plant physiology is easy to understand compared to plant anatomy. In plant anatomy, you have a lot of things to remember. Alright, some things related to plant reproduction also I have considered. So I have considered plant physiology and plant reproduction. If there are questions which will talk about comparative analysis between different divisions of the plant kingdom, whether it is in terms of physiology, anatomy or reproduction, I have skipped it for now. Alright, for that we will have to build on the understanding of different divisions of the plant kingdom. So let us keep our focus narrow in understanding plants. Alright, once you understand plants, it is easy to understand and easy to recall, get the questions right than human biology. Alright, but we don't generally get good answers to the plant related questions because the terminologies are not relatable. We don't relate to those terminologies. So in our attempt, we will have to correlate. All living organisms are similar. All organisms are similar, whether it is plant, humans or bacteria. Any of the living organisms, they all have similar functions. Purpose overlaps. Every organism wants to survive, every organism wants to reproduce. That is the purpose. Design is based on where it is, how it survives, its environment, its ecosystem that it survives in, and its evolution. The design is based on that. Based on where it survives, where it has to survive and what are the conditions for reproduction, different organisms will have different designs for different, that's all. Function also will be dependent on the design. Function is to ensure that the purpose is achieved with the given design. That's all, no? So it is the same for all organisms. Understanding life in totality is easier. We will keep humans as a reference to understand everything. To understand all life forms, we will keep humans as the reference because it is us. We can relate to what we are and correlate it to others. Alright, that is the basis on which we will be doing. And post-test analysis is based on a lot of questioning. You have to question, you have to ask questions which are common and general in nature. Not going into too much techniques. The simpler you keep your questions, the questions being related to curiosity, better will be your analysis. You will see how it can be done now, okay? I have done it for the plant physiology questions. And in every paper I am giving basis of how many questions have appeared from plant physiology and human physiology. I have told you, as we keep analyzing this, you will see plant physiology and human biology will cover almost 30% of the paper for you. 30% of 90 questions. So 20 to 30 questions. In the initial days of NEET, this is mock NEET 1 is 2013 NEET paper. So then there were a lot of questions related to examples. As we keep moving forward, by now you will be attempting 2018 paper now, no? The sixth one, 13, 14, 15, 16, 17, 18. You will be attempting 2018 paper on Monday, tomorrow. You will see the number of questions that are related to examples. How many of these have a particular type of estimation? How many of these have a particular ovary? Such questions are reducing in number. More questions related to concepts are increasing. So, analysis of the paper is important. In this paper, 2013 NEET paper, the total number of questions that were there in plant physiology were 9. Alright, so we'll keep moving. Question number 95 is the first one that was related to plant physiology. Megasporangium is equivalent to embryo sac, fruit, nucellus, ovule. This question is completely a lot of terms related to plants. There is megasporangium, nucellus, embryo sac, fruit, ovule. 5 terms are there. The first thing that should come to your mind is, what are these? What are these? Where do we find them? What is its purpose? How is it structured? When we understand that for all of these terms, we will be able to clearly understand what megasporangium is, and the answer will come out. So what is megasporangium? Just random thought, whatever, you have to keep it simple. What is megasporangium? And there will be so many related topics that we will have to understand to get an answer to this question. All right. I will run through, okay. You have the training in post-test analysis is the training for asking questions. Those who know how to ask questions are the ones who will get answers. Those who focus on remembering answers, ready-made answers, are the ones who will fail. All right. So post-test analysis is extremely important. However hard you may find it, how much ever struggle you go through doing it, how much ever blank you draw while you are attempting it, it's all fine. But once you start getting a hang of it, you will know how to ask questions. All right. Asking questions is the most important ability for knowledge building, for analysis. All right. Okay. So two things are there here. You have embryo sac and ovule. Embryo sac is a part of ovule. Nucellus is also a part of ovule. But embryo sac is having a specific reproductive function within ovule, which has other reproductive and non-reproductive functions also. Correct. So we need to understand the structure of both. This diagram you will know. What is the diagram? What is this diagram? This is a diagram of? What is this diagram of? This is a diagram of? Embryo sac. This is ovule. Inside the ovule we have the embryo sac. So embryo sac is inside the ovule, and it is having so many things surrounding the ovule. So many structures are surrounding the embryo sac in the ovule. So ovule is a complex structure. Okay, let it be. We will see. What is this? This is embryo sac. Embryo sac. So there are so many things here: antipodals, synergids, egg, filiform apparatus, polar nuclei, central cell. So many structures are there. How many cells are there in an embryo sac? 7 cells. How many nuclei? 8 nuclei. How many haploid cells? How many haploid cells? 6 haploid cells. How many diploid cells? 1 diploid cell. Does the central cell have 2 nuclei fused or separate? Central cell will have 2 nuclei fused. It will be a central cell with a diploid nucleus. Why is it then referred to like separate cells here? Just to make you understand, there are 2 cells of the same nature coming together, which will fuse and eventually become the central cell. All right. Okay, let it be. We'll move. Then megaspore, what is it? There are, see, in the life cycle of plants, we will look at 2 stages repeatedly: gametophytic stage and sporophytic stage. Gametophyte, phyta means body. Plant body is referred to as phyta, P-H-Y-T-A. That is why the most undifferentiated plant division is thallophyta, then bryophyta, pteridophyta. Then we have gymnospermae, angiospermae. Why not phyta for gymnospermae and angiospermae? The reference is not to the body there. The reference is to the type of reproduction they will have. Both of them are producing seeds. Spermae, seed. Gymnospermae, naked seeds. Angiospermae, covered seeds. That is the differentiation. That is the reference to the division there. But thallophyta, bryophyta, pteridophyta, the reference is to the body. Thallophytes, undifferentiated body. Thallus is the body of fungus. Thallus is the body of fungus, undifferentiated body. That is thallus. Fungus-like body is thallophyta. Bryophyta, slightly differentiated but no clearly formed root and shoot system. Bryophyta. Pteridophyta, slightly advanced than bryophyta, root and shoot is formed, but no seed formation. Gymnosperms, root and shoot well differentiated, naked seeds. Angiosperms, root, shoot well differentiated, enclosed seeds. That's all, no? So that is the design. Purpose is the same for everybody. Root is to anchor and draw nutrients. Shoot is to provide stability and produce food. Right? Based on the design. Purpose is that. Purpose is root should provide anchorage and it should provide mechanisms for absorbing nutrients. Now there is no root in Thallophyta. How do they provide anchorage? How do they fix? How does it draw nutrients? The body is so simple. The body is attached to the surface like fungus body. So the entire body functions in pulling out nutrients from the soil, from wherever they are, predominantly water. Correct? Go to the bryophytes. Because they develop small rhizoids, root-like structures, they have a possibility of going inside. Not very deep, but to a certain extent. They can hold on to something. So, the shoot also can grow a little bit. Do they grow very big? They can't grow very big because how tall you grow is dependent on how deep you exist. Applicable to human beings also, no? Right? How far you grow in life depends on how deep you understand life. Plants, similar. So they can go a little deep, they can hold on, they can provide anchorage, but they cannot go deep, therefore they cannot grow tall. Mechanisms, how do they derive nutrition, how do they derive their essential substances from the surroundings other than carbon dioxide from the atmosphere? There are mechanisms, we will see that. Pteridophytes, root and shoot formed. Vascular tissue in pteridophytes. Do they have vascular tissue? Yes, they do. So vascular tissue provides mechanism for transportation in the body of the plant. So the plant can grow. They can grow above the surface to a certain extent. But they cannot grow very tall. Why? The roots are not very deep. So pteridophytes grow to a certain extent, bigger than the bryophytes, but they cannot grow into trees. Gymnosperms advanced further. They became better. They became better at survival. They became better at their ability to grow. But they developed the ability to ensure the survival of their reproductive propagules. What is a reproductive propagule? Something which is used for propagation. Spores are reproductive propagules. Some plants will produce spores. Fungus will produce spores. What happens? Those are cells which have the ability to produce a new body. Spore can be considered as something that has the ability to produce a new body. Seeds can be considered as equivalent to spores. Will all the cells in the spore be capable of producing a new body? There will be certain cells which are capable of transforming into a new body. Most of the others are for supporting it and protecting it. Alright. When you are just throwing away your gametes and the reproductive cells into the atmosphere, into the surrounding has lot of things which can destroy it. So whatever you are, whoever you are, whether a bacteria or a fungus or a plant or a human, you have to design mechanisms to ensure that the rate of success of reproduction is higher. So that will determine how many gametes are produced. See, sperms are produced in millions. Ovum is produced in single digits. Why? Ovum is fully protected. It's like Z plus secure, not Z plus. It is like NSG commandos. Nobody can just reach the ovum. Ovum is well protected. It doesn't even walk. It is carried. It is carried in the fallopian tube, no? So protective mechanisms. How do you protect it? How do you ensure that the success is increased? Survival, reproduction comes down to that. What increases your survival in the surroundings that you exist? What increases your reproductive possibility in the surroundings that you exist? In the surroundings that you want to reproduce. Those lead to mechanisms of alteration. Now this fellow, embryo sac, is designed to protect the gametes. Embryo sac is designed to protect the female gamete, which is the egg cell inside the embryo sac. So there are so many things around it. Why all of these are there, we will see. Alright, next as we go down. So we need to understand megaspore, gametophyte, sporophyte. You see, gametophyte is the reproductive stage, sporophyte is the growth stage. Then integuments. What are integuments? Protective layers. Integuments. In human body, what are integuments? This is integument. Nails are integument. Integuments are hard protective structures. Alright. Then what do you find in a seed as you go from outside to inside, or an ovule? You find integuments, you find micropyle, you find nucellus, you find embryo sac, and you find funiculus. What are each of these and what do they do? We will go one by one. So you see, when you are doing post-test analysis, you will start from where the question is and go to related concepts which will help you gain mastery over the concepts that are related to the question. Alright. So then we go further, ask some more questions. What is megasporangium? Where do you find it? What is its purpose? What is the design and function? What can be considered as equivalent to megasporangium? Equivalent structures. There are confusions there. Should we consider the entire ovule as megasporangium, or should we consider the nucellus as megasporangium? That we will have to make a judgment call, because the entire ovule develops from the sporogenous tissue in the plant body, and that sporogenous tissue also differentiates to form megasporangium. Megasporangium gives rise to megaspore mother cells also, and the undifferentiated megasporangium cells will become the nucellus, which will be the protective layer. The differentiated megaspore mother cells will become the embryo sac. Now both the embryo sac and the nucellus are coming from megasporangium tissue only. What should we consider as the right choice? Should we call the entire ovule to be equivalent to megasporangium, or should we consider the nucellus to be equivalent to megasporangium? That is a judgment call that you will have to take in this question. Appears very simple, but there are things that you have to think and answer. Some people might, if you see objections to the questions in 2000 paper, this would have been an objection also raised by somebody. Ovule also comes from megasporangium, the entire ovule comes from the megasporangium tissue. Differentiated megasporangium gives rise to megaspore mother cells. That goes on further dividing and becoming embryo sac. So embryo sac is coming from the megaspore mother cells. Nucellus is undifferentiated megasporangium. What should we consider as the right choice? The explanation would have been, nucellus is closer to what megasporangium is than embryo sac, because embryo sac is in the sequence of differentiation. As you differentiate further, you are becoming more specific. What is closer to its structure resembling megaspore? For that we need to understand what is the structure. Fruit, purpose, design, function. Nucellus, purpose, design, function. Ovule, purpose, design, function. Alright. So this is the notes that you have to make for one question. Alright. So how many chapters you touched upon, how many concepts you touched upon? I will read out. Let us assume you have done this exercise and you have answered the questions. I will not explain, I will just read out the answers to the questions. Then we will see the level of understanding that you get, and then I will take it up again for explanation. Would that be okay? Alright. And while making notes, it should not be just a copy paste from your textbook or reference material. It has to be a simple English representation of what the concept is, what it is. Initial papers, you will have to do extensive notes, but as you keep going further, any question that comes related to the same concept, you don't have to redo the notes, you already have it in your notes. So as you keep moving forward, the number of questions for which you will have to make an analysis, post-test analysis notes will reduce. Alright. And you will see that the same concepts are giving you questions repeatedly. Same concepts give you questions repeatedly in different dimensions. So if a question appears in a different dimension, you have to address that dimension. You don't have to go for the entire explanation. The ovule, if you see, there are two ends, callosal end, callosa end, and micropyle end. Micropyle is an opening. Micropyle is an opening ideally suited for the entry of pollen tube. Pollen tube through which the pollen grain comes and the male gamete enters. The male gamete is supposed to reach here, reach the egg. But it is not necessary that the pollen tube enters through this only. The pollen tube can enter through here also. The pollen tube can enter laterally also. Based on that, we have different ways of pollen tube entry. That we will see when the question comes. Alright. Then if you see from outside, there are two layers of covering. Correct? What are these layers? Coverings are referred to by the term integument. There is an outer covering and an inner covering for the embryo sac. Leave it here. The coverings are not differentiated here in the funicle. Funicle is the bridge, you can say, a bridge connecting the ovule to the placenta. Placenta is drawing nutrients from the plant body. Like placenta in human beings. Placenta is connected to the mother. Placenta is the junction between the mother and the developing baby. So placenta draws or whatever is essential from the mother and delivers it to the baby. Similarly here also, there is a placenta which is designed to deliver what is essential for the growing ovule, taking it from the plant. Alright. Now coverings are done. What is this? This tissue surrounding, that is the around the embryo sac. What do you find around the embryo sac? That is the nucellus. Nucellus is haploid, diploid, triploid. What is the ploidy? It is the undifferentiated megasporangium, diploid. Embryo sac is formed after meiosis in the megaspore mother cells. So this entire assembly is the embryo sac. In the entire assembly of embryo sacs, you have antipodal cells. What is its role? Antipodal cells provides polarity to the embryo sac. On one side you will have this, here you will also have filiform apparatus. Filiform apparatus and the antipodal cells will act as the guiding structures for the pollen tube to reach the egg. The pollen tube has to reach the egg, no? So this is the embryo sac. You have 7 cells, 3 plus 3 plus 1. This fellow secondary nucleus is diploid. Secondary nucleus is diploid. Will we find a cell enclosing the secondary nucleus in the embryo sac? We said the embryo sac has 7 cells. We can see 6 cells and a nucleus. Where is the cell there? Endosperm is triploid. Embryo is diploid. What if it is a tetraploid plant? What if it is a tetraploid plant? Tetraploid plant means, what is the ploidy of a tetraploid plant? Ploidy is 4. If it is a tetraploid plant, what will be the ploidy of endosperm? 5. That is where, see, you know embryos, the cells of the embryo are diploid because we are considering a diploid plant. You know if the cells of the embryo are diploid, the endosperm has to be triploid because we have seen here 2 haploid nucleus will fuse with the haploid nucleus of the micro embryocyte or the male gamete to form a triploid cell. What if the plant is a tetraploid cell plant? Question will be like this only. There will be a story. So and so plant is tetraploid produced by so and so technique and all the distractions. The endosperm of the seed produced in this plant will be diploid, triploid, tetraploid, hexaploid, or not even diploid, triploid, tetraploid, pentaploid, hexaploid, because somebody already told 5. We will give one for that confusion also. You know what the embryo sac is, you know the ploidy of the cells in the embryo sac, you know the ploidy of the embryonic cells and the endosperm cells are not the same. You should be able to answer this question now. A tetraploid plant is produced by using the principles of biotechnology. The purpose of doing this tetraploid plant was to increase productivity of the yield of this plant. What will be the ploidy of the endosperm of the seed in this plant? The options will be triploid, tetraploid, pentaploid, hexaploid. Huh? Hexaploid. Why? How many of you agree that it is hexaploid? Raise your hands. If you agree it is hexaploid, raise your hands. Okay. Raise your hands. Okay. How many of you disagree? No, it cannot be hexaploid. See, when it is a yes-no question, do you agree or disagree is a yes-no question. For a yes-no question, there are 5 options. Alright, for a yes-no question, there are 5 options in my school. You have to choose one of the options. The answer, she says, is hexaploid. The endosperm of a tetraploid plant will be hexaploid. How many of you say yes? Raise your hands. Okay. How many of you say no? Raise your hands. How many of you say don't know? Raise your hands. Oh, okay. How many of you say might be this or that? I'm not sure. Can't say. Raise your hands. Can't say. You're confused. How many of you say the gangsters of the class? I won't say. Okay. So 5 options for a yes/no question. Yes, no, don't know, can't say, won't say. Anybody who wants to choose the 5th option, won't say, raise your hands. Okay. So some of you are gathering half courage to do that, because you are not yet sure that you can be a bigger gangster than me. So once you are confident about that, you will start choosing the 5th option. I won't say. What will you do? You can't do anything. Okay, I'll rephrase the question. A particular plant is a diploid plant and the seed was examined for ploidy. The embryo and the seed was examined for ploidy. This was an endospermous seed which had endosperm. What will be the ploidy of the endosperm? Haploid, diploid, triploid, tetraploid. Now diploid plant, what will be the ploidy? Huh? Triploid. You have no confusion with a diploid plant having a triploid endosperm. But if the ploidy of the plant becomes different, can the ploidy of a surviving organism be odd? Can it be an odd number? For a sexually reproducing organism, can the ploidy be odd number? Can a sexually reproducing organism be triploid? Can it be? Not possible. Why? Because a sexually reproducing organism has to divide the ploidy for reproduction. It is not just reducing the numbers into half, it is reducing the ploidy into half. How will you divide the ploidy? Complete sets of chromosomes when the number of sets of chromosomes is odd. If you have 5 sets of chromosomes, one cell will have 3 sets, another set will have 2 sets. So the next generation will not have the same ploidy. So a sexually reproducing organism, unless it is an aberration, should have even ploidy. That is clear? If a question comes which will tell you that the ploidy of a sexually reproducing organism is some odd number, and they will talk to you about the ploidy of the gamete, there will be an option, there will be one of the options which says data incorrect or wrong, something which will tell you that the question is wrong. All right? So you should not ignore that. You should not always a question will have some answer. There will be questions which cannot have an answer. Question being wrong, it cannot have a right answer. Question is wrong is the right answer. All right. Now, look at it in a different way. For a diploid plant, egg is haploid. It is a product of meiosis. Egg becomes haploid. The male gamete becomes haploid. They both fuse to form a diploid cell, zygote, which will develop to produce the plant. Clear? Always think about ploidy when it comes to reproduction in terms of fraction and not exact numbers. For a diploid plant, 2x or 2 ploidy is the full ploidy, correct? What will the gamete have? The gamete will have half ploidy. Don't say haploid, half ploidy. Central cell is formed by combination of 2 half ploidies. When the male gamete comes, the male gamete is also half ploidy. When the male gamete fuses with the central cell, what will be the ploidy of the central cell? 3 half ploidies, correct? The central cell is, the fertilized central cell which becomes the endosperm is 3 half ploidy. So half ploidy becomes full ploidy. The central cell is 3 half ploidy. So if the full ploidy is diploid, what is half ploidy? If the full ploidy is diploid, what is half ploidy? Haploid. The endosperm is 3 half ploidy. You combine 3 haploids, what will it be? Triploid. Now if the plant is tetraploid, what is half ploidy? If the plant is tetraploid, what is half ploidy? 2, diploid. Endosperm will be 3 half ploidies. Endosperm is 3 half ploidies. So 3 half ploidies in a tetraploid plant would be 6 hexaploid. All right. Questions that will lead you to wrong answers when it comes to ploidy, endosperm, embryo sac, these things would be that small lack of understanding of ploidy. You will remember it as diploid, haploid, endosperm being triploid because you read that. But what is necessary to understand is full ploid, half ploid. The sporophytic generation is full ploidy. The gametophytic generation, the gametes are half ploidy. Now whatever is the ploidy of the parent plant, that is the full ploidy. Half of that is the half ploidy. Instead of telling haploid, talk about it in terms of half ploid. When you look at ploidy in terms of half ploidy, you will be conscious about what is the full ploidy. You understand? So you can also look at the endosperm as being 0.5 ploidy, 0.5 ploidy, 0.5 ploidy. That is 1.5 ploidy. 3 half ploidy is 1.5 times the full ploidy. That's all, no? So what is the ploidy of the endosperm of a given plant is dependent on what the ploidy of the plant is. If the ploidy of the plant is diploid, endosperm is, you have to keep talking. The more you talk, the more faster I can go. All right. I am not like a cinema actor who acts in front of the camera. I am like a theater artist who acts based on the response of the audience. The more responsive the audience is, the more faster the story goes. Otherwise, I'll keep waiting for responses. So if you have understood, the response is whatever you have understood. If you have not understood, the response is you have not understood. Okay? There should be more noise. I am not used to, and I don't like silent classrooms. I like aggressive classrooms, not going to the side of violent classrooms. Just aggressive classrooms in answering. Okay, if the ploidy is diploid, if the full ploidy is diploid, the ploidy of endosperm is triploid. If the full ploidy is tetraploid, the ploidy of endosperm is hexaploid. If the ploidy of the plant is 6x, the endosperm would be? The endosperm would be? Eight. 9. Why? It is 1.5 times the original ploidy. You get it? So that is what you need to understand here. So with this, we know what is the ploid. Assume this is the ovule of a tetraploid plant. What is the ploidy of this? If it is a tetraploid plant, what is the ploidy of these synergids? Diploid. What is the ploidy of the egg cell? Diploid. Okay. This one, antipodals? Diploid. Central nucleus or secondary nucleus? Tetraploid. Tetraploid. After fertilization, this will become hexaploid. It can be a diagram. Now you have seen that the central nucleus after fertilization becomes 1.5 times the ploidy. Overexcited about it. Question is asking you about secondary nucleus ploidy. Question can be, the ploidy of the secondary nucleus in an ovary will be the same as the ploidy of option A, megaspore mother cell, option B, sporophytic cells, option C, megasporangium cells, option D, all of the above. Same thing. It is the same concept. Question can be different. What would be the option? What would be the right choice? The ploidy of the central nucleus of the ovule of a plant will be the same as option A, megaspore mother cells, option B, megasporangium cells, option C, sporophytic cells, option D, all of the above. All of the above. Why? All of them are full ploid cells, diploid cells. All of them are full ploid cells. None of them are cells which are product of meiosis. If the question is, the ploidy of the central nucleus of an ovary of a plant cannot be the same as megaspore mother cells, sporophytic cell, lenticell, option D, none of the above. The ploidy of the central cell or the ploidy of the secondary nucleus of an ovary of a plant cannot be the same as option A, megaspore mother cell, option B, sporophytic cell, option C, lenticell, option D, none of the above. All of them have same ploidy. Okay, let us assume the plant is diploid. None of the above means all of the above. Correct, no? There is a negation in the question. None of the above is a negation in the choice option. Negation becomes assertion. The question can be reversed as no, it has to be all of the above, not none of the above. If the question was the ploidy of the central nucleus of the ovary of a plant is the same as, then the three options and the fourth one would be none of the above, because none of the above has the same ploidy as that of the central cell. Lenticell, where do you find lenticell? In this paper only you have one question on lenticell. Alright, lenticell is also diploid. We will see when we go there. You see this? Some people talk about biology being a subject where you have to just memorize. Huh? There is nothing which can give you success at a higher level by just memorizing things. Everything at a lower level can operate with just remembering things. Anything at a higher level requires the same level of mental acumen. Analysis, logic, application, correlation, interpretation, all of this is required for everything. You understand? Questions can be double negation questions. None, okay. Which of the following is not so many things? One of the option is none of the above. Then the question can be rephrased and reread as which of the following is all of the above. It's the same. Alright? And this cannot be taught. I cannot teach you this. Concepts can be told, you can understand it, but logic evolves. Logic should evolve by analytical application of your mind, trying to push the limits of your understanding. Alright? See, one question. We are stuck here. I have not even gone to further analysis of this question. This is how you go about. When you are reading something, some other question should crop up in your mind. You go find out an answer to that question, then come back. Something else will crop up. Go find out. But the anchor for your intellectual exploration will be this question. The question that you are analyzing is the anchor for exploration of multiple things. So you are starting from here, going somewhere, coming back, going somewhere else. From there you find something, you go somewhere else, but you pull back to the same question. You see the building of a spider's web. Alright? Okay. So this one, let us keep it now. For now, enough. We will explore the same concept in a different dimension later. So what all we could understand by analyzing this? What is this for? What is this for? Placenta? Nourishment. Acts as a bridge between the mother plant and the developing embryo or the ovule. This is a bridge to hold this entire assembly. Coverings. Why do we need coverings? Embryo sac has delicate cells. Embryo sac has cells which are delicate. Egg cell is there. You need to protect it. To protect it. Not only protection during the before fertilization, also protection after embryonic development. Alright. So integuments will become seed coat. The outer integument will become the outer seed coat. The inner integument will become the inner seed coat. So that will also become a fibrous tissue which will protect the seed. And nucellus will become what? Nucellus will be used up during embryonic development. The endosperm will develop into the endosperm will develop from the central cell. The endosperm will develop from the central cell. Endosperm is the reserve food material for embryonic development post seed dispersal. Fruit is also for that only. Why do plants produce fruits? Two purposes for fruit. Can you tell me? If you are a plant, your job is to ensure you produce seeds. Seed has endosperm which will provide food for the growing embryo when it is inside the soil. Because as long as it doesn't come out, it cannot do photosynthesis. From the time it goes into the soil till the time it comes out and start producing green structures, chlorophyll laden structures, the young stems and leaves, it will not be able to produce food. How will it get energy for doing that process of coming out from inside? That is taken care of by endosperm. Endosperm is like the plant giving endosperm to the seed is like your mother giving you a bag of food when you are going on a trek. Alright? Or you packing up some food while going uphill. You will not find anything on your route to the en route to the peak until you come back. You need to have enough food to help you survive till you come back and get access to food. Same for plants. They need to carry something. What is the purpose of fruit? Fruit will make the seed heavy. How will it help in dispersal? So it is a trade. Plant is doing business with animals. Plants are doing business with animals. Plant is telling the animals, I'll give you something to eat. Don't eat here. Take it away. It's a takeaway restaurant. Okay. Why doesn't the plant want the animal to eat the fruit there? In fact, plants will prefer other animals eating their fruit than human beings. Why? Build some logic around it. If you are a plant, you would rather give your fruit to other animals, maybe a cow or a goat or a giraffe or an elephant, but not human beings. Why? Two reasons. Why? Think like a plant. No. Be louder. See, one reason is plants will eat the fruit along with the seed. So they don't remove the seed and eat, no. And plants will take the animals will eat the fruit along with the seed, and they keep moving. So by the time the seed comes out, the animal would have gone somewhere else. The plant does not want its babies to grow around it. It's only human beings who want the babies to grow around them, and babies to stay with them till 30 years, 40 years, sometimes lifetime. All right. Plants just tell their babies, I have given you enough things, get lost, grow wherever you want to grow if you can survive. All right. So animals will carry the seeds, they eat the fruit. They have motivation to eat the fruit. They don't have the dexterity to remove the seeds, they eat it along with the seed. Seed is provided with a lot of hard coating. Now as the seed passes through the gut of the animal, the coating becomes softened. It improves the possibility of the embryo to grow. The warmth of the gut of the animal would stimulate the embryonic development process. So the time the seed has to spend inside the soil is reduced. The animals will give out the seed in some other place along with some other reserve material for the seed to eat and grow. Correct, no? The feces of the animal is food for the growing plant. So it is not only eating the fruit. The animals are doing more good to the plant by eating the fruit than the plant did to the animal. The animal would have even eaten leaves and got its energy. But plant is taking so much effort. It is designing the fruit so well. Mango with different aroma, jackfruit, triggering the bears to climb big trees, open it and enjoy it in a party. Bear party has a lot of jackfruit and honey. All right. So plant does all that. Animals also benefit. They also get variety of food. Like us, animals also need different varieties of food. They carry the seed far away, disperse it along with two benefits for the seed's further development: softening the seed coats, making the embryo to develop faster, initiating the process of embryonic development as the seed passes through its gut, and also it will not wash the seed and throw it, no. It will just drop it along with its feces. So manure also for the plant to grow. What we do, we take the fruit, we sit under the tree, eat the fruit, throw the seed there only and go. Plant is like, we are like freeloaders. Okay? We are like freeloaders who go and pick up whatever we want to eat and don't even pay money. We are like the street gundas who will go to these small shops. They will take what they want don't pay money. And throw the wrappers and all the waste there only. Okay. So you see this fruit and all its advantages. So anyways, this is enough. That's what I tell. If you start analyzing one question properly, this one question is enough for us to cover entire biology, all the chapters. You should know how to build the links. Alright, but we will not do that. We will go question by question and expand it little by little. We have, good, this is very good. We have 258 more days. You have 8 times more time than what you have already spent in the last 30 days. Alright. So many things we analyze. Now again I'll ask you, what is the ploidy of polar nucleus? Assume the plant is diploid. What is the ploidy of polar nucleus? What is the ploidy of polar nucleus? Haploid. Why? It is not yet fused to form the central nucleus. See, this is for reference. I will not give my notes to you in print or soft copy until October 31st. And I will give it to only those people who will show me their completed notes. Alright. You should show your completed notes. All the post-test analysis up to date. Post-test analysis, even if you have completed 60 to 70% of the papers by October 31st, I will still give it to you, because after October 31st, you will not have any other notes to make, only post-test analysis notes to make. Even if you are lagging behind in the post-test analysis notes making, it's okay. Do the analysis properly. Don't be in a hurry to complete post-test analysis in 1 week. Let's say you have completed 2 papers. 3 papers are pending. Don't jump to the current paper. Do analysis in a sequence only. It's okay, you go back to the paper you attempted 3 weeks ago. It is still fine, but do it in a sequence. As you keep doing it, the pace will improve. If you are sincere, I am sure you will not be having a gap of more than 30 to 40% on the post-test analysis by October 31st. If you are not sincere about it, or if you keep doubting, if you keep questioning, things won't work. No matter how much you doubt, no matter how much you question, this will remain the same. The process will remain as I want it, because I know it works. You better believe it works and keep working according to plan. Then it will work for you. There are 2 ways of realization. Realize and do things. Easier, that requires you to do a lot of study, lot of understanding, analysis and all that. Second easy thing to do is do and then realize. Do and then realize, because who is telling you to do it has already done it multiple times for various exams for thousands of students, and there is realization of how it works and why it works and how it builds your internal connections and all. So just like you believe there is a God who will save you, believe that this process will work and stick to it. Alright. But keep track with your linear notes. Syllabus map is done, level 1 notes, do it properly, completely. By October 31st, all the perspective based notes also should be complete. Your lag should not be more than 15 to 20% by October 31st. If you are completing the linear notes by October 31st, you are in a good position. Even if you have 30 to 40% lag on post-test analysis, because after October 31st, the frequency of test will become once in 4 days. Then you will have more papers getting accumulated. But by then, if you are sincere, you again have 2.5 months, more than 2 months, about 50 days now, about 70 days. If you are sincere in doing it, you would have got the hang of it. You would know how to do it. All right. Don't expect to feel comfortable every day. This process has made you uncomfortable. It has pushed you to the limits of your comfort zone. It's good. That is how growth happens. Only when you expand your comfort zone, you become better. Playing within the comfort zone will not help in growth. All right. So I'll just read now. What are megaspores? Large reproductive cells. That's all. It's a simple explanation for it. Megaspores are the cells in plants that eventually become the female part of the plant body. Okay. How do we make it appear as one page at a time? Not one page, spread it out. This one, huh? No, but for this, is this visible? The size? No. In the last, the size of the text is not visible. You are not able to read? No. Now you can read. Megaspores are the cells in plants that eventually develop into the female part of the plant where the egg is formed. So, which is the part which will lead to the development of egg? So, by this we will know megaspore is that part of the plant body which will eventually lead to the formation of embryo sac, the ovary and all, ovule and all that. All right. What is gametophyte? In the life cycle, we will have two stages, gametophytic stage and sporophytic stage. So, gametophyte is reproductive stage. The gametophyte is the stage in a plant's life cycle that produces sex cells or sperm and egg. Sporophyte is the growth stage. Sporophyte is the stage in a plant's life cycle that grows and produces spores, which can eventually lead to the formation of new plants. Now, are the spores diploid or haploid? Can I consider zygote to be a spore? Is zygote part of the gametophytic life cycle or sporophytic life cycle? Zygote is the first stage in the sporophytic life cycle, correct? So whenever we talk about spores, we should know spores when dispersed will lead to production of the new organism of the same kind. What are integuments? Protective layers. Can I sit, please? Integuments are protective layers. Integuments are the layers that surround and protect the seed inside the ovule of a plant. That's all. Where are they coming from? What do the integuments lead to? Seed coats. Integuments are developing from what? They are developing from the nucellus tissue only. Alright. So integuments, protective layers surrounding the ovule. In the ovule, what all do we see? Integuments, micropyle, small opening where pollen tube enters, nucellus tissue that nourishes the developing embryo sac, female gametophyte that contains the egg cell. Alright. Funiculus, stalk that attaches the ovule to the ovary wall. What is the structure on the ovary wall to which the funiculus is attached? What is the structure on the ovary wall to which the funiculus is attached? Placenta. Alright. So what is megasporangium? Megasporangium is a structure found in seed plants where megaspores, which are a key part of the female reproductive organ, and eventually, where megaspores are formed, it is a key structure of the female reproductive organ and eventually gives rise to female gametophytes. Now, gametophyte is something which will contain the gamete. In the ovule, what is the gametophyte? What can we refer to as gametophyte? Embryo sac. In the ovule, we can refer to embryo sac as the gametophyte. Location. Where will we find the megasporangium? Found within the ovule of flowering plants. It is enclosed by integuments and is commonly referred to as the nucellus. That is the question, no? What is megasporangium equivalent to? But I told you there is a confusion between nucellus and the entire ovary. Not ovary, the entire ovule. Purpose, the main function of the megasporangium is to produce and protect the megaspore mother cell, which undergoes meiosis to form megasporangium or megasporocyte. Megasporocyte is the embryo sac. So, by this we can understand there is megasporangium, gives rise to megaspore mother cells. The rest of the cells will remain as megasporangium tissue. They will grow around this megaspore mother cell, differentiating megaspore mother cell. Megaspore mother cells undergoes meiosis to produce how many cells? 8 cells. Alright, to produce 8 cells. And that continues further. One of the megaspores develop into the female gametophyte or the embryo sac. One develops into the embryo sac. Correct? 3 cycles. See, after meiosis, how many cells do you have? You will have 4 cells. 3 of them degenerate. One will undergo 3 more cycles of mitosis. One will undergo 3 more cycles of mitosis to produce 8 haploid cells. That is the megaspore development. So, can you tell me the sequence? Can you tell me the sequence from megasporangium to embryo sac, to the embryo sac? Megasporangium is the first one. Then, next. Megaspore, mother cell. Then, megasporocytes. How many? 4 megasporocytes. In the arrow mark you can represent meiosis. Megaspore mother cell to megasporocytes, meiosis. Then in the 4 megasporocytes you can write 1 surviving megasporocyte. 3 degenerate in the arrow mark. Then 3 cycles of mitosis. 8 nucleus. Then formation of 8 cell with 8 nucleus. Formation of cell membranes around, then migration of nuclei. 3 nuclei, 2 in the center. Then formation of cell membranes. That is the embryo sac. Correct, no? So this sequence you should remember. Questions can be jumbling up the sequence. Jumbling up the sequence can be question. Stages of the sequence can be question. Alright, function of each of the stage, ploidy, all of these can be questions. Process is the same. You understand the process completely. Whatever be the question, how does it matter? You have to think, correlate it with your understanding, map it to something, arrive at the answer. Alright. So, one basic principle that you can apply while doing post-test analysis for biology is PDF. What is the purpose? What is the design? What is the function? This can be the starting point. If there are terms, you have to know what each one means. If there are interrelated terms, correlating terms, you have to understand the connection between each of those terms. If there are terms related to different concepts, you have to think why those have become a part of the options. While designing questions for the exam, it is always taken care of to ensure that even the wrong choices will have some connection to the question. That is a good question. So you have to think, why are these given as options? So if one of these is to become an option that somebody chooses, where in the logic they might have gone wrong to arrive at this particular place as the answer. These are the things you should do. So 3-point agenda. Three-point agenda while doing post-test analysis. Number 1, go with the PDF approach. Purpose, design, function. This will help you understand most of the things. Even let's say Southern blotting technique. Southern blotting technique or PCR, polymerase chain reaction. You can interpret that also in the same way. What is the purpose? Why do you want to do it? How do you design so that the purpose is achieved? And the function. What is the process which is followed to ensure that the purpose is achieved? What is PCR done for? Polymerase chain reaction is done for what? Amplification of DNA, amplification of DNA. That is the purpose. Why do you want to do it? Why do you want to amplify DNA? To study the DNA. To study the DNA for further analysis. Do you do PCR for every sample? You will do PCR when the sample that you have is very small in quantity. If you have an entire limb of a person, will you do PCR? To study DNA of the person? Do you want to do PCR? If you have an entire limb of the person, you will still do. You will pick up a small tissue, amplify it and do it. But technically you don't have to do it. But if you have a hair follicle, if you have a hair follicle that you find in a crime scene, do you want to do a PCR? You want to do a PCR because the amount of DNA that you find in that sample will not be enough to do an analysis. So you do PCR. So these kind of questions you need to ask in common laymanish terms, like a layman, like somebody on the street asking you questions. That is how your clarity comes. Okay. Done, no? Design and function. Megasporangium is typically surrounded by protective layers called integuments, which later develop into the seed coat. Function, it houses the megaspore mother cell that undergoes meiosis to produce megaspores. Out of 4 megaspores formed, usually 1 megaspore becomes functional and develops into the female gametophyte or embryo sac. This ensures that the female gametophyte is well protected and nurtured during development. That's all. Simple language. Whatever is the language that you are comfortable with, English, let the flow of the explanation be in that language, be in that kind of expression. There is nothing which is right way of making notes or wrong way of making notes. How you put it should be the way you talk, the way you understand, the way you speak English. Simple. It can be grammatically wrong, but if it is helping you to understand it better, write it. It's not a literature competition, no? Alright. Notes is supposed to be your reference material for quick recall. If somebody else has made it, it will not be useful for you. Like textbook. Somebody else has made textbook, no? Why are you not able to quickly gather information from that? Because it's not in the way you talk. It should be in the way you talk. Purpose, design and functions of embryo sac, fruit, nucellus, ovary. Now you tell me, we will apply the same purpose, design, function for embryo sac, nucellus, fruit and ovary. What is the purpose of embryo sac? Purpose of embryo sac? Okay. Eventually formation of the embryo. Then? Speak. What else? Don't look at the board. Look inside. Look in your own mind. Get answers from your mind. There is nothing there that you have to repeat. What is the purpose of embryo sac? Reproduction. Develop and nurture female gamete. That's all, no? Enough. Design. Design. Produced as a 8 nucleus 7 celled structure. Provides polarity for the entry of pollen tube. Provides tissue that will eventually develop into endosperm for the nourishment of the developing embryo. Function, this process, how it formed and how it further develops into embryo, that will go into the next phase of discussion. That's all over. Then nucellus, purpose of nucellus, protecting the embryo sac, provide nourishment to the developing embryo, contribute to the formation of coverings which will help in the protection of seed eventually. Design, remains diploid or remains full ploid, present around the embryo sac, function, draws nourishment from the placenta, vascular tissue from the placenta, and delivers it to the embryo sac, contributes to the development of the embryo sac into embryo and endosperm during the development of the seed. That's all, no? Over. Fruit, purpose, dispersal of seed by providing food for animals, then protect the seed, protection of the seed and dispersal of the seed. These are the two purposes of producing fruit. Design, based on what the kind of seeds that you have to distribute, you will have seeds in the form, fruits in the form of fleshy fruits or dry fruits, various types of fruits based on what is the purpose of the plant. Function, same, overlaps. All right. So purpose, design, function. Okay, done. This is the analysis for one question. How much time did we take? 1.5 hours, 1 hour, 15 minutes mostly. All right, got a fair idea? So now you understand it takes time to make post-test analysis notes, right? And I have discussed it, so it has taken so much time. If you are looking around in books, your NCERT books, you should be able to get it quickly. Should not take so much time. It will not go so deep also when you are doing it. It's okay, whatever is the depth you can achieve. But ask more and more questions. All right. Next question is question number 98. Interfascicular cambium develops from the cells of medullary race, xylem parenchyma, endodermis, pericycle. There are different structures that are listed here. Cambium, fascicle, interfascicle, medullary race, parenchyma, endodermis, pericycle. All of these, are they present only in the root or shoot, or present in the root? Only in root or shoot, or both? All these structures. Now let us list down the terms. What do you mean by a fascicle? Fascicle, F-A-S-C-I-C-L-E, fascicle. Why are you doing this? Previous year paper, sir. Looking at solutions. What do you mean by fascicle? Those are the questions we need to ask. There are terminologies. What is the meaning of each of the terms is what we have to understand. Forget about the concepts, just the terms. Wherever you get fascicle, the meaning will still be the same. Bundles. Fascicle means bundles. Bundles of something similar. Okay. Can I say an axonal fascicle? Bundle of axons, can I say fascicles? Can I say a nerve is a fascicle of axons or dendrons or both? Can we say that, no? Bundle. Wherever you find fascicle, it means bundle. Inter means what? I-N-T-E-R. Inter and intra are the two terms you have to remember as contrast. What do you mean by inter? Between. Intra means within. So interfascicular means what? Between bundles. Interfascicular means something that is in between bundles. Between bundles of what? Not mentioned here. Interfascicular what is mentioned? What is between fascicles now? In this question, what are they talking about? What is between the bundles? Cambium. What is cambium? What is cambium? Sorry. I have developed a condition called BPPV, benign paroxysmal postural vertigo. Inner ear problem. I forgot to take the medication in the morning. It leads to a lot of giddiness. So incoherent speech. There are calcium crystals in the inner ear, and there are semicircular canals. You know that, no? Semicircular canals are for what? Position sense. There is fluid inside the semicircular canal which will give you a sense of position. Disturbance of the fluid is what will cause giddiness. That giddiness is referred to as vertigo. In the condition that I refer to, BPPV, for various reasons, the calcium crystals in the inner ear gets displaced and they get into the semicircular canals. Now these calcium crystals are thicker, heavier in density than the fluid. So positional change will lead to these calcium crystals moving at a different pace than the fluid in the semicircular canals, leading to disturbance in the signals that are sent to the brain about position. So change of position will lead to vertigo. So I have to move around like a zombie, straight, not change too much. It will resolve by itself. It will resolve by itself, but long-term medication. I am on vestibular sedative. Vestibular sedatives are the drugs which will sedate the vestibular apparatus. Vestibular apparatus is where? It's in the inner ear. All right. Now, what is there between the bundles in this question? The question is referring to cambium that is in between bundles. What is cambium? In plants, we will find two types of tissues based on their ability to divide. What are the two types of tissue that we refer to based on their ability to divide? Meristematic tissue and permanent tissue. Meristematic tissue becoming permanent tissue is referred to as differentiation. Correct? It is referred to as differentiation. Is the reverse possible? Can permanent tissue become meristematic tissue? Yes or no? Those of you who say yes, raise your hands. Permanent tissue also can become meristematic tissue. Raise your hands if you say yes. Okay, those who say no, raise your hands. No. Once permanent, always permanent. They cannot become meristematic. Okay. Those who say don't know. Oh. Can't say. Won't say. Now those of you who did not choose an option, stand up. You are choosing an option now or standing up? Choose? Okay. Okay, sit. Now I'll again ask you, those of you who say permanent tissue also can become meristematic tissue, raise your hands. Now more hands are coming up. Those who say no, raise your hands. No. Don't know. Can't say. I feel it may, but I also feel it can't. Okay. Meristematic tissue becoming permanent tissue is referred to as differentiation. Permanent tissue becoming meristematic tissue is referred to as de-differentiation. Now this de-differentiated meristematic tissue again becoming permanent tissue is referred to as re-differentiation. Three terms you have to remember here. Alright. Cambium is meristematic tissue which will add girth to the plant body, which will contribute in the secondary growth of the plants. Cambium. Alright, cambium is basically meristematic tissue. Wherever they are, based on the position, they are referred to by different names. Alright. So differentiation, de-differentiation, re-differentiation can be correlated like this. I am a non-alcoholic. I start consuming alcohol and I become alcoholic. That is differentiation. My wife gets to know about it, kicks me hard, and I become non-alcoholic. This is de-differentiation, addiction, de-addiction. All right. Now, after I got de-addicted, after I stopped taking alcohol, my wife continued to taunt me, referring to my earlier habit of alcohol. So I said, get lost. I will again start drinking alcohol. Redifferentiation. All right. So addiction, de-addiction, re-addiction. And cambium which are produced by de-differentiated cells, de-differentiated meristems. We'll see that. Cambium you understand, which will divide and add more cells to the plant body. So we understood what is fascicle. What do you mean by intrafascicle? Intrafascicle means in between the bundles. There is interfascicular cambium. There is intrafascicular cambium also, that is called vascular cambium. Between xylem and phloem there is vascular cambium. All right. What is medulla? In a plant body, the outer part, outside to the endodermis, is referred to as cortex. The part inner to the endodermis is referred to as medulla. You consider two concentric circles. One big circle, that is the boundary of the plant body, epidermis. One small circle in the center, the circumference of this circle is the endodermis. The space between the outer circle and the inner circle is the cortex. The space inside of the inner circle is the medulla. That's all. Okay. Medullary race. What do you mean by race? See, wherever you get cortex and medulla, you should understand that. We find cortex and medulla in the kidney also. Cortex is something on the outside. Medulla is something on the inside. What is the boundary is what you have to see in every place where you find cortex and medulla. Okay. So, yeah. Race means what? Something that is radiating. Something that is radiating. Medullary race might mean what? You need not know anything. If you know what is medulla and if you understand what is race, you can make a guess on what medullary race can be. Something that is radiating from the medulla. Clear? Enough. Let it be for now. Xylem parenchyma. Xylem means what? Xylem is the water conducting tubes in the plant body. Network of cells and tubes which conduct water. That is xylem. Network of cells and tubes that will conduct food is phloem. That's all. Plants require two transportation mechanisms, one to transport water, one to transport food. Very simple mechanisms they have. Parenchyma means? Parenchymatous tissue is a simple tissue which acts as supporting tissue. It does not have strength. Three types of simple meristems are there. Simple meristematic tissue. Parenchyma, collenchyma and sclerenchyma. Parenchyma, collenchyma and sclerenchyma. Based on their shape, their structure. Based on where they are present. At the tip you have apical meristem. In between you have intercalary meristem. In the vascular tissues you have vascular meristem. That's all. Alright. The advantage of concepts in biology is you understand the meaning of terminologies. They will have stories. They will tell you what it is. You don't have to memorize the meaning and then correlate the name. You just have to know what the name means. So, xylem parenchyma, the supportive storage oriented tissue around xylem, that is xylem parenchyma. Endodermis, what is the endodermis? What is endodermis? I told you, no? The circumference of the inner circle. Endodermis is that layer of cells which will separate the xylem and, which will separate the cortex and medulla. Endodermis is the layer of cells where the cortex ends and the medulla begins, if you are coming from outside. That is the place where the medulla ends and cortex begins, if you are going from inside out. That's all. What is pericycle? Pericycle. Pericycle is another layer of cells that are found around the endodermis, next to the endodermis, in the inner layer, inner part of the plant body. That's all, no? So, we will see. Each one we will explore now. Sequence of cells or tissues from outside to inside in a stem. If you are going from outside to inside of a stem, what all do you encounter and what each one means? Epidermis, outermost protective layer. See, if you ask right kind of questions, you will have answers to all this. In your NCRT, you have all this. You just have to ask it. Cortex, layer of cells between the epidermis and the vascular bundles. Endodermis, innermost layer of the cortex which regulates the flow of matter into the vascular bundles. Pericycle, a layer of cells just inside the endodermis that can give rise to lateral roots. Pericycle is the structure from which lateral roots can be produced. Okay, vascular bundles, xylem and phloem. Xylem on the inside and phloem on the outside, responsible for the transport of water, nutrients and food. Pith, central part of the stem, often used for storage. So tell me the sequence of tissues that you will find. Let's say we are going into the stem. What is the first thing that we will encounter? Epidermis. Then we go into cortex. The end of epidermis is arrived at when we reach endodermis. Immediately after that we have the layer of pericycle. Peri means what? Around. Cycle means all around. Cycle, where it starts, it ends. So pericycle is not present in one part of the plant body. If you take a cross section, it is all around it. It's like a cycle. Then as you go inside further, you will find vascular tissue. Xylem on the inside, phloem on the outside. At the center of the stem, you will have pith. Pith is for storage. What do we find as we go inside the root? Epidermis, same. Cortex, endodermis, pericycle, vascular cylinder. The arrangement of vascular bundles in the stem and roots is different. That we will see. When we get a question where we have to deal with the structure of these, when we are dealing with anatomy questions, we'll see that. Alright. Difference between the root and stem. One important difference. Pith is not found in roots. Pith is found only in stem. Pith is found only in stem. Question can be, pith in the center of the structure, in the center, at the center is not found in A, monocot stem, B, monocot root, C, dicot stem, D, dicot root. Option A, monocot stem and monocot root. Option B, dicot stem and dicot root. Option C, monocot stem, monocot root. Option D, monocot root, dicot root. Option D, because it is not found in either of them, both of them. Both dicot and monocot, pith is not present in roots. But when the question starting monocot, stem and root, the options will start confusing you. When we do this question, if you clearly put it into your mind that pith is absent generally in roots, they might be present in some roots but insignificant, you will be able to make a clear choice. And you should know that whether it is monocots or dicots, pith is present in the stem. So then anything with stem is ruled out. Even if you are doubtful whether the pith may be present or absent in monocot or dicot root, you know it is definitely present in stem. So all three options have stem in one or the other two, one or the two options. Monocot stem, dicot stem, monocot root, dicot stem, some combinations like that. All right. What is cambium? Cambium is a layer of actively dividing cells between the xylem and the phloem in plants. It is related to the secondary growth of stems and roots, leading to an increase in thickness. Can we consider the primary growth of plant as being responsible, as being a consequence of cambium activity? Cambium is a general reference to secondary growth. Okay, we have intrafascicular cambium and we have intrafascicular cambium, two types. Medullary rays are also cambium. Purpose, design and function of cambium. The purpose of the cambium's primary purpose is new cells for the plant, contributing to the plant's growth in thickness. Growth in thickness is secondary growth. Cambium is designed as a thin layer of meristematic cells that lie between the xylem and phloem tissues. This structure allows it to efficiently produce new cells, adding layers to both the xylem and phloem. On the outside it keeps adding phloem tissue, on the inside it keeps adding xylem tissue. Function: produces secondary xylem on the inside and secondary phloem on the outside. It also plays a key role in the healing of wounds and the formation of callus tissue in plants. What is callus? Callus is a plug. Wherever the vascular tissue is damaged, you have to seal it to prevent leakage of vascular fluid from the tubes, from the structures. So callus is a mass of cells that will seal the damage. Callus is found in something else. We refer to something else as callus in another concept. Where else do you get callus? Tissue culture. In tissue culture, you will find callus. You take what is the process that is happening in the formation of callus? You are taking maybe a stem, a small stem, initiating the process of tissue culture, and you form that, you develop callus. What happened in the stem? Option A, differentiation. Option B, dedifferentiation. Option C, redifferentiation. Option D, none of the above. What happened in the stem that you have taken for, or what happened in the stalk that you have taken for tissue culture when it forms callus? What happened? Differentiation, huh? Yeah. Come on, what is there? What is it? Why is it so difficult to correlate? Differentiation. So the meristematic cells became permanent cells, while callus got formed. For tissue culture, do you want meristematic cells or permanent cells? You have to first transform the permanent cells into meristematic cells, that is callus formation, and you have to start initiating the growth and differentiation in the callus, which will lead to the formation of root and shoot. Then you will saplings and take it through the process of hardening, and then you will transfer it to the field. So callus formation is a consequence of what? Callus formation is a consequence of dedifferentiation. Permanent tissue, that is the stem piece, that is the stem stalk that you have taken, is becoming meristematic tissue. Then it starts producing continuously. You will get unlimited supply of callus. That's the advantage of tissue culture, no? You can take any permanent tissue from any plant and initiate the tissue culture. You will have unlimited stock of callus. You can take that callus, part of that callus, into another chamber and start initiating the process of differentiation, root and shoot development. What are the two hormones that you will use in combination to bring about root and shoot formation? Auxin and cytokinin. Auxin and cytokinin. Increase one, shoot differentiation. If you increase the other, root differentiation will happen. All right. Okay. So what are the different types of cambium? Cambium also can be referred to by different names. We will have the ones which are related to this particular question. Primary types of cambium: vascular cambium and cork cambium. Vascular cambium and cork cambium. Now here There is cork, bark, phellem, phellogen, phelloderm, so many terms, confusing terms. We will look at it when the time comes, but just know phellogen is a reference to cork cambium. Phelloderm is a reference to bark. Anyways, we'll see that later. Types of cambium. I have given a table. Types of cambium, origin and function. Vascular cambium develops from the procambium in the vascular bundles or from the dedifferentiation of mature parenchyma cells in the interfascicular region. So, vascular cambium is produced from what? Procambium in the vascular bundles, between the bundles. So, vascular cambium, is it intrafascicular cambium or interfascicular cambium? Is it interfascicular cambium or intrafascicular cambium? Intra? Vascular cambium has two things. See, you have one bundle, one vascular bundle here, another bundle here, another here, another here, goes on like that all around. You have parenchyma cells in between the vascular bundles. Between xylem and phloem, in the bundle, there is cells that will transform into vascular cambium. The ring of cambium should get completed, no, for the growth to happen. So between the bundle, within the bundles, there is intrafascicular cambium. Between the bundles, to continue this ring, cells adjacent to the two intrafascicular cambium will dedifferentiate to become intrafascicular cambium. So it is both interfascicular cambium and intrafascicular cambium. Interfascicular, intrafascicular, intrafascicular, interfascicular, goes around like that. So by the end of it, you have an entire ring of cambium. You understand? So wherever the intrafascicular cambium is contributing to cells, it will lead to phloem production on the outside, xylem production on the inside. The ring where the interfascicular cambium is, when it divides and produces more cells, it will lead to increase in the parenchyma cells. Both should happen, no? It's procambium, primary meristem. Primary meristem. Interfascicular cambium is secondary meristem. Alright. The intrafascicular is coming from the procambium. It is not dedifferentiation. Dedifferentiation is the interfascicular cambium. Now he is one vascular bundle. I am one vascular bundle. This is phloem. This is xylem. Where is the vascular cambium? Here. I have one. You also come. Here we have, let's say at the level of the shoulder, we have intrafascicular cambium. You stand here. Where are you facing? Stand face forward. He is also one vascular bundle. Who is he? Parenchyma cell in between the two bundles, medullary cells. Now, I already have my cambium. This is the cambium, my shoulder. He is permanent tissue now. He is not dedifferentiated. He is not meristematic yet. So this portion of his body will become dedifferentiated. Now, this is next to each other. Come, stand. There is already cambium here. There is cambium there. Now cambium has to be produced here. So here what happens? Here what happens? Dedifferentiation. Dedifferentiation happens. Dedifferentiation happens in the intervening interbundle tissue. He is the tissue in between the vascular bundles. Correct, no? So there is already meristem here. Intrafascicular cambium, procambium, vascular cambium. But imagine people standing all around. One, if I am the cambium, he is the if I am the vascular bundle, he is the parenchyma tissue in between the vascular bundle, medullary tissue. He is the vascular bundle. Next again there is parenchyma in between. Again there is vascular bundle, parenchyma, vascular bundle, parenchyma, vascular bundle, goes on like that, no? So the cells adjacent to the cambium in the vascular bundle will de-differentiate and form the continuity between the two vascular bundles cambium. Go. You understand? That is how a ring is formed. Now when that ring is formed, all of us will start adding more and more people to our line. So I will bring more people in front of me and behind me. Or here, we are talking about this way, no? This is phloem and this is xylem. I will add more cells above and below. Same with him also. If that does not happen with the cells in between, will the shape of the tissue remain the same? This will grow tall, this will remain short, this will grow tall, that will remain short. The shape will get distorted. So to maintain the shape, what should happen? This also should multiply. So together the ring is adding cells on the outside, on the inside, increasing the girth of the plant body as a whole. That is why you need this interfascicular cambium. Cork cambium arises from the outer layers of the cortex in stems and roots, produces cork on the outside and secondary cortex, phelloderm on the inside, contributing to the formation of protective bark. Bark is phelloderm, phloem, phellogen. Phelloderm, phellogen, phloem. Phelloderm is the bark, is the cork. Phloem is the cork, phellogen is the cork cambium. Phloem is the secondary cortex. Okay. So outside you have the hard, dead part of the bark. That is the bark outside. Cork is outside the bark. In between you have the cambium, which is adding cells on the outside and inside. What is going outside is dying. What is going inside is contributing to the formation of secondary cortex. That we will see later. Alright, but for now, just understand cork cambium and vascular cambium. Both of these are contributing to increase in the girth of the plant body. Is that clear? See, all of this you will find in NCERT. You just have to ask the right questions and look at the right places in the NCERT book. Now, we had so many structures mentioned in the question. I like writing tables which will compare the things. So all of it we need to know, no? Medullary rays, radial sheets or ribbons extending vertically through the stem. From the endodermis, from the pericycle, the cells which go into the core of the are medullary rays, derived from the vascular cambium, location in the stem and roots running horizontally. Okay. Purpose, transport of water, nutrients and food across the plant stem. Medullary rays will help in transporting food across the plant stem. Now there is one vascular bundle, there is another vascular bundle. Somebody should give water and food to these cells in between, no? The medullary rays will help in doing that transportation mechanism. Design and function, designed as horizontal sheets, they facilitate lateral transport and store nutrients within the plant. Same with xylem parenchyma, derived from vascular cambium. Within the xylem tissue they are present. Storage of water and nutrients, lateral transport of water. You do this. Endodermis, innermost layer of the cortex, derived from the ground meristem. Ground meristem is primary meristem or secondary meristem? Huh? Ground meristem is primary meristem or secondary meristem? Primary meristem. It forms initially only, no? Endodermis. Okay, then we have pericycle, a layer of cells just inside the endodermis. We have already discussed that. Derived from the procambium. Procambium is also primary meristem. Surrounds the vascular bundles in roots, give rise to lateral roots. Pericycle gives rise to lateral roots. Lateral roots are coming from the pericycle. You need to know that. It is not coming. Questions have come where the options will be epidermis, endodermis, pericycle, cortex, all four. So lateral roots are not attachments on the surface. They are in growths coming from the inside. So it's not easy to pull away. If you pull the lateral roots, you will still see a small stump inside of the lateral root. Alright. So this is with this question. What is the answer to the question? Interfascicular cambium develops from the cells of medullary race, xylem parenchyma, endodermis, pericycle. Medullary race. The cells of the medullary race will undergo dash to give rise to interfascicular cambium. Fill in the blank. The cells of the medullary race undergo dash to give rise to interfascicular cambium. What will be that dash be? What will be the dash? Options: differentiation, de-differentiation, redifferentiation, any of the above depending on the climate. De-differentiation, no? Yeah. It's not any of the above depending on the climate. Okay. Alright. So don't bother about what the right answer is. Even before you look at the answer keys also you can do analysis. Don't bother about what the right answer is. Do the analysis and then you will arrive at the right answer. Then you can cross check with the answer key. That is also possible. But you need to look at the answer keys beforehand for you to note it down. Alright. Ideally, if you are doing your self preparation, you have to do you have to document the the scores after you do the post test analysis, not before. But now you do the scoring, document it in your table and then do the post test analysis, no problem. Alright. Where do we not find pith? Roots. Lateral roots develop from pericycle. Cambium that develops from the medullary race is called interfascicular cambium. Endodermis develops from which cambium? Endodermis develops from procambium. Phylogen is primary cambium or secondary cambium? Phylogen is secondary cambium. De-differentiated cambium is not primary cambium or secondary cambium. De-differentiated cambium is not primary cambium, secondary cambium. Primary cambium, because de-differentiated cambium is secondary cambium. That's all. Vascular cambium is present outside the xylem. True or false? Vascular cambium is present outside the xylem. True or false? Vascular cambium is present between xylem and phloem. True or false? Vascular cambium is present between xylem and phloem. True or false? False, huh? Where is it present? Within the? What do you mean between xylem and phloem? Okay. See, the same thing. What we discussed so far is the basis of all these questions. My primary job is to expose you all and tell you that you can overcome the limitations and show you the path to overcoming limitations. Inner to the phloem in the vascular bundle. True or false? True. That's all. Okay. If you try to recall answers, you will never be ready for any test. If you learn to think and arrive at answers, No test is outside of your preparation. All right. So here, to answer the same, to answer multiple questions related to the same topics that we discussed, not all the topics, only one, positioning of the cambium, is the only thing that I used to create some 5-6 questions. You have to go back to the basic understanding of the concept, try to imagine that, picture that in your mind, see where is the thing that you are being asked present, and then with respect to the question, answer. All right. Intrafascicular cambium and interfascicular cambium are not the same, are not similar in terms of its origin. True or false? Intrafascicular cambium and interfascicular cambium are not similar in terms of its origin. True? Explain. Explain. Answer is correct. Intrafascicular cambium and interfascicular cambium are not similar in terms of its origin, though they are similar in terms of its functions. Why are they not similar in terms of the origin? Intrafascicular cambium is procambium or primary cambium, not procambium, primary cambium. Interfascicular cambium is secondary cambium. Okay? Interfascicular cambium has not undergone differentiation yet. It remains meristematic. Interfascicular cambium became meristematic after it was a permanent tissue. So interfascicular cambium is produced by dedifferentiation. All right. So, okay, we can stay here exploring this, that, just think. So when you complete all 180 questions in 1.5 hours and you want to get out of the class, even if you don't get the answer, spend the entire time till the last minute, 1:45 to 5:20. The entire time you should sit here, even if you don't know the answers, try and make sense of the questions. Understand the questions in such a way that you will be able to ask the question in your mother tongue. You should be able to translate the question in your mother tongue. No need to know the answers. If you spend the entire time of the duration of the test to understand the questions also, that is still much better than getting out of the classroom, getting out of the room during the examination. All right. So don't ignore even the smallest of things. Everything to be followed as it is. The slightest of deviation, imagine it will transform into exponential losses. Slightest of deviations. Okay. You don't understand it right now, but stick to bit of instruction. Every bit of instruction. No deviation whatsoever. Subject will grow. Subject will improve. But what will destroy you is deviation from the process. If you stick to the process, your subject will improve, because sticking to the process will make you stay committed for 11.5 hours every day for 258 more days. If you are sincere in observing what's going on within yourselves, you should have by now 32 days of presence here in this process. Tiredness, daytime sleepiness should have come down by now. You should be able to do the Surya Namaskaras and all the physical exercise routine in the morning much more comfortably. You are not doing all that, all those sounds should have come down. All right. Restlessness would have gone down, because 30 days of de-addiction from your mobile phones. Things will only keep getting better. Don't keep cribbing about what you are going through. Enjoy and embrace. It's painful. Discipline is painful. Mind doesn't like it. But if you willingly learn to enjoy it and embrace it, if you accept it, it will keep on improving. Right? One thing that I spoke with some parents are in continuous touch. All right. Some of you have had health issues. Some of you are experiencing sleeplessness. Parents are raising that, sir, my child is not getting enough sleep. Tell them how to sleep. All right. Okay. And about all the junk food that you are getting, we have instructed your parents to not supply you with junk foods. So next time your parents will bring you loads of fruits, dry fruits, nuts and all, energy bars, which will go as nourishment to the plants, is it? One of my cousins did not like green leafy vegetables, milk and all. But when we used to go to our aunt's place, she will forcefully give us a big glass of milk. I enjoy it, my sister enjoys it, my cousin, she doesn't enjoy it, she doesn't like it. She takes the glass of milk, it's a big house. She goes into the balcony, there are some plants, she will dig some soil, pour the milk. Just to not get caught, she will distribute the milk to 4 or 5 plants. And she will just cover it with mud again. And once she was given green leafy vegetables to eat, green leaves to eat, she did not like it. She threw it on the attic. After about 2 months, when some cleaning was being done, there is some green color distortion on the attic. The leaves and all had become fungated, it had dried up, it has become powdery, but the wall had green mark of it. See, every crime will have its evidence. All right. However intelligent the criminal is, there will be some evidence left. Then further analysis of that led to who the potential criminal might be. The one who can go to this room, looking at the recentness it is recently, so who all has come in the last 2 months, who can freely enter this room. It will narrow down. And then you know the history of the people who have visited. You know it is this person exactly who has done it. Anyways, don't do that. So stick to the process. That's what I wanted to tell you. All right. We'll go to the next question. Cambium. We are done. This paper has glycolysis, Krebs cycle, electron transport chain. We will not be able to touch it today. We'll do it. All the 3. How many of you are comfortable with glycolysis, Krebs cycle and electron transport chain? Raise your hands. You are comfortable with it? So many questions can come from that. Which about the enzymes, about the energy dynamics, about the reversibility of the reactions, about the rate limitation. Phosphofructokinase is the rate limiting enzyme of glycolysis. Why is it so? Why NADPH should be considered equivalent to 3 ATP? Why FADH2 is equal to 2 ATP? Why will you die if you don't have oxygen? Such questions can be answered by understanding respiration in all 3. So this is done. We have successfully completed how many questions? 2 questions. But do you realize how many concepts we have touched upon? So many concepts. That's the advantage of doing learning by post-test analysis. You have an anchor to build the knowledge. Right. Age of tree can be estimated by its height and girth, biomass, number of annual rings, diameter of its heartwood. Easy question to answer, but there are concepts to understand. What is the answer to this question? Number of annual rings. So if you just want the answer, you just see it's a number of annual rings and go away. But there are a few things that you need to understand. Height and girth. What is height? For human beings it is clear. Heel to crown. Heel to crown is the height for human beings. For plants, height is what is above the ground or the entire plant from the root till the tip. height of the plant is the entire length of the plant, both root and shoot put together, or is it only the shoot? I'm sure I don't have to ask you only the root, because that will be a very easy response to give. Should we consider the entire length of the plant to talk about its height, or should we consider only the shoot? What should we consider? Only the shoot or both the shoot and root? Both the shoot and root? Only the shoot. You consider the height of the plant from the ground level to the tip, to the highest point. Okay, done. Girth means the circumference of the plant. Where do you count? Where do you measure it? At the base of the plant? Where do you measure it? There should be standardization, no? It is referred to as the breast level of the plant or the chest level of the plant. Basically the mid length. At the mid length you measure the girth of the plant. Girth is the circumference, diameter. I mean circumference of the plant. How big is the circumference of the plant? That is the girth. What is annual ring? So there are some things here. Annual ring. Annual ring has two types of wood produced. Spring wood and autumn wood. Spring wood, autumn wood. So we need to, with this question we have to understand what is annual ring, what is spring wood, what is autumn wood, why is the, how does it appear? Is spring wood lighter or darker? Same with autumn wood. What is heartwood? What is sapwood? Which one should we consider while we are calculating the annual rings? Should we consider the number of rings formed by the heartwood or the sapwood or both? It is not just enough to know. We measure, we find out the height of no, age of the plant by counting the annual rings. Not enough. Let us see. Height, measurement of how tall something is from the base to the top. In trees, central wood of a tree, which is okay. Circumference is we have dealt with that. Heartwood. See, okay. Heartwood is made up of xylem only. True or false? Heartwood is made up of xylem only. Heartwood is made up of xylem only. True or false? There is vascular cambium. It will add more and more xylem to the inside, more and more phloem to the outside. So phloem keeps getting pushed outwards, xylem keeps getting pushed inwards. Even in the xylem, the number of vascular bundles, the diameter of each vascular bundle is dependent on how much water is available. If water is available in abundance, do you have bigger vessels, fewer in number, or do you want to have smaller vessels, larger in number? So that will tell you the difference between spring wood and autumn wood. In spring, do you have more availability of water or do you have more availability of water in autumn? Spring. Rainy season corresponds to spring or autumn? Autumn. Isn't spring February, March, that is before your summer starts? So spring wood is formed when the availability of water is not so high compared to autumn wood. You need more vessels to conduct water when there is more availability or less availability. more availability. So in the same space, you will have more water conducting tubes produced during autumn. So autumn wood will be darker in color. Spring wood will be lighter in color. It goes on like that. So one band of darker ring and one band of lighter ring, both constitute 1 year. 2 important seasons are taken care of. That is one annual ring. Alright, it is xylem, water conducting tissue. Now heartwood is what is to the inner side of it. Inner side of the vascular cambium got compressed, or it becomes more and more filled with resins and gum and other substances, becomes harder and darker. That is the hardwood. Sapwood is also called softwood. That is still carrying out the function of conduction. It is still conducting water. The xylem that is there, softwood xylem, that is still conducting water. Phloem becomes a part of the outer softwood. Phloem is not considered as the wood as such. Phloem is the ring that will be removed off when you are using the wood for any commercial purposes. Alright. So we will see each one of those. Those are the questions that we have to look at. Hardwood, heartwood or hardwood, sapwood or sapwood. Sapwood is the outer layer of the wood in the trunk just beneath the bark. In trees, the living part of the tree that conduct water and nutrients from the roots to the leaves, that is a part of softwood. Annual ring. Rings visible in the cross section of a tree trunk representing 1 year's growth. In trees, each ring marks a year of growth with wider rings indicating more growth during favorable years. So if you count it backwards, you can even date the years. Which year was this ring? If you find a particular ring more thicker, that means that year this plant had got more water. The conditions were favorable. So you can go back to the weather data, rainfall data, and correlate it, cross-check it, whether during that year there was adequate rainfall. If the annual ring is small in a particular year, that means that during that year the rainfall was not adequate. So the ring did not become big enough. It was small. Okay. So it gives you correlation with the weather also, with the rainfall data. What is heartwood? Heartwood is the central non-living part of a tree trunk, usually darker, harder and more resistant to decay than the outer layers. Where does it come from? Heartwood forms from the older sapwood as the tree grows. Over time, the inner layers of sapwood no longer conduct water and become heartwood. So it is not separately formed. Initially, every xylem tissue is conducting tissue only. As more and more xylem is being formed, the inner layers will start getting deposits of resin, gum and other chemicals, blocking these tubes. The tubes get blocked. It provides strength, but the problem is it no longer can conduct. So because of the deposits, it will start becoming darker and harder. Sapwood, whatever is still conducting, it is considered as sapwood. Heartwood was also earlier sapwood. Sapwood eventually becomes heartwood. So there is always a layer of wood that is conducting. There is wood that is everything else inside is not conducting, adding to the strength. How big and enormous the tree can grow is dependent on the size of this heartwood. It will provide strength to the tree. Why this is done, no? Why should it be produced, or what is its purpose? Heartwood provides structural support to the plant and makes it strong. Trees, it is dense and strong, helping the trees to stay upright and resist external forces like wind and gravity. That is why it is produced. Heartwood is designed with dense, tightly packed cells that are often filled with resins, oils and other compounds that make it resistant to decay. It will not decay so easily. It is protected. It keeps the tree strong. Function. The primary function of heartwood is to support the tree structurally. It no longer plays a role in the transport of water and nutrients, which is the job of the sapwood. Conducting water and nutrients is the job of the sapwood. It is not of the heartwood. Okay. Does it provide any disadvantage to the plant? That is also a question that I am asking you. What disadvantage? While heartwood provides strength, it no longer contributes to the tree's growth or nutrient transport. It is just there. It doesn't contribute to growth or transportation. Also, once it becomes heartwood, it is more prone to fungal infections if it becomes exposed. Why you should not puncture the trees with big nails? You puncture the trees with big nails, the nail can reach to the heartwood, and you pull out the nail, it becomes a conduit for fungus to go inside. Fungus gets a hard base on which it can grow, and it is also having access to the sapwood where it can derive nutrients from. So they can start gradually decaying the wood from inside. What are annual rings? Concentric circles visible in a cross section of the tree trunk formed by the growth of new wood each year. How do we count the rings to determine the age of trees? Method. To determine the age of a tree, you count the number of annual rings visible in a cross section of the trunk. Each ring represents 1 year of growth, with the outermost ring indicating the most recent year. It determines the age of a tree by counting its rings. You count all the rings that are in the cross section of the tree trunk, which includes both the sapwood and hardwood rings. This is another question that will be explored in exams. Annual rings we understood. Heartwood is the hard one which is on the inside. Should we count sapwood also while counting annual rings? Should we count sapwood rings also? Sapwood is also producing rings, no? Which one will be darker, heartwood or sapwood? Heartwood will be darker in appearance. Heartwood is harder in structure. Sapwood is lighter in appearance because it is still conducting, so it has hollow interiors. But do we count sapwood also to determine the age of the plant? Should we count sapwood also? We should count sapwood also. These are the things that you have to be aware of. These can be, what is heartwood, what is sapwood, what do you do to count annual age? Count the annual rings, done, over. But if it is taken to the next level of questioning, where understanding is questioned, you should know this. Sapwood is also counted. Now tell me, will the thickness of heartwood be the same as it was when it was? Will the thickness of heartwood, the ring, be the same as it was when it was sapwood? We know that all wood, whether it is even the heartwood, was sapwood once upon a time. Once upon a time it was sapwood. When it becomes heartwood, will the thickness of the ring be still the same, or will it be lesser, or will it be more? Those of you who say heartwood thickness, the ring thickness in the heartwood will be the same as it was when it was sapwood, raise your hands. Nobody thinks that. Okay. Those of you who think, no, heartwood would be compressed, so its thickness will be lesser than what it was during its existence as sapwood. Raise your hands. Okay. Those of you who think the thickness of heartwood will increase than what it was during sapwood, raise your hands. Okay. What we witnessed now is a feature of democracy. Okay. The right choice is the option for which nobody voted. Two other choices for which votes were cast are the wrong choices. The thickness of heartwood remains the same what it was when it was sapwood. The difference is Initially when it was sapwood, it was conducting water and nutrients, but later it starts getting deposits. Because of deposits, it became hard, not because of compression. It did not stop conducting water and nutrients because it got squeezed. No. It stopped conducting water and nutrients because it started getting deposits. It started producing deposits inside, blocking it. Okay. If this room is one vessel, which is a sapwood right now, there will be sand and rocks that are getting deposited into this room. And eventually, nobody can move around in this room. So it became hardwood, harder. The room did not get compressed. You did not press the room and bring the walls closer to each other so that made nobody walk through it. No. Clear? So these are the nuances that you have to be aware of. Annual rings, count it, you will know the age of the plant. Everybody will answer that. The challenge is, if there is something else that you need to answer which can be answered only on understanding, most of the students drop out. A pharmacist gives medicines without prescription in our country, unfortunately. Pharmacists are responsible for a lot of antibiotic resistance in our country. Cost of treatment being increased is significantly because of the pharmacists giving antibiotics without doctor's consultation, without doctor's advice. Anyways, no problem. A pharmacist gives the same medicines. You go to a doctor, the doctor checks you, the doctor says nothing to worry. You are having high grade fever, you go to the doctor, you go to a doctor, doctor will examine you and tell you, you will be alright in 2-3 days, it's a simple viral infection, no need to worry. Take paracetamol 650 mg 3 times a day for 5 days. And the doctor takes 500 rupees consultation. You go to the pharmacist, you take 15 tablets of paracetamol 650 mg which will cost you 20 rupees. The pharmacist will give you some special advice. I could have only given you this. Why did you waste 500 rupees to the doctor? Isn't it? Why did you waste 500 rupees to get only paracetamol? You could have only done it at home. You pay the doctor for the doctor telling you nothing is wrong. Okay. Otherwise, fever can be the symptom of a simple viral infection to cancer. Fever is a symptom, it is not a disease, no? It requires an expert to tell you it is not cancer, it is just simple viral infection. You can take paracetamol. What if it turns out to be something else? Correct? By the time a person completes 5 days of paracetamol, he might be dead. Then that 500 rupees would have led you in the direction of recovery, not in the direction of mortuary. Alright. So, deeper understanding is required to solve problems which common people will not be able to solve. Deeper understanding does not require you to read huge books. It requires you to understand the foundational principles properly. If you understand the basics, deeper understanding is not in advanced concepts. Deeper understanding is in the strong fundamentals. If your foundation is strong, you have a deeper understanding. Advanced analysis can be made individually. Effort is required to strengthen the foundation. Deeper, complex calculations will emerge out of your understanding. Alright? That is why the linear notes that you are making is slowly empowering you to make your foundation strong. Alright? Parallelly, post-test analysis is making you develop the ability to analyze. Okay? So, this is vary. No, I already told you this. And I also explained this growth process. Annual rings are formed each year. They become hard because of deposits and not because of compression. That should be clear. This transition does not change the thickness of the rings that were already formed. Okay, they are not compressed and broken down and sandwiched. The thickness of each annual ring is determined by the growing conditions during that year, such as the availability of water, nutrients and sunlight. Favorable conditions lead to thicker rings while unfavorable conditions result in thinner rings. Basic things. Heartwood is simply the older non-functional wood that no longer transport water and nutrients, usually darker and more resistant to decay, but they themselves remain in the same width as they were originally formed. Alright. Sapwood, younger functional wood that is actively involved in transport. So done. Our question came now. 108. Okay. This itself will require an entire session. We will be discussing in detail all process of glycolysis, Krebs cycle and electron transport chain, everything that you need to understand it and be able to answer whatever be the question. After the session you should be able to remember also. Remember the entire pathway. How many reactions are there in glycolysis? 10. How many are there in Krebs cycle? 8. How many are there in, what is there in between? Glycolysis, oxidative decarboxylation and Krebs cycle. What are all these for? I will only give you the basic outline of this entire process. We will discuss the details later, but before that you tell me the answer. 3 boxes in this diagram represent 3 major arrow represent net reactants or products. 4, 8 and 12. What are they? 4, 8 and 12 can be what? Okay, tell me what pathways are these? Pathway A is what? Pathway A is what? Pathway A is glycolysis. Pathway B is what? Krebs cycle. Pathway C is electron transport chain or oxidative phosphorylation. There are 3 types of phosphorylation. Phosphorylation is where you phosphorylate ADP to produce ATP. 3 types of phosphorylation is found in living systems. One is oxidative phosphorylation, where you transfer the energy in some high energy compound in an oxidative process to ATP. Happens in mitochondria. Second one is that happens during photosynthesis. What do you call that? Photophosphorylation. Third one is what happens during glycolysis, Krebs cycle. We find ATP production in glycolysis and Krebs cycle also, no? That is also phosphorylation. What phosphorylation is that? Substrate level phosphorylation. 3 types. When a substrate is being acted upon, energy that is released is used to phosphorylate ADP to make ATP. Substrate level phosphorylation happening at the level of substrate, not requiring any specialism, not happening in the mitochondria. Oxidative phosphorylation is use the energy of high energy compounds to be transferred to ATP production, transferred to ATP, phosphorylated with the phosphate, produce ATP, happens in mitochondria. Requires a specialized mechanism to deliver this. Photophosphorylation is the same thing happening in plants by the influence of sunlight. Utilizing the energy of sunlight fixed in certain complexes to produce ATP. No reaction in a living organism can happen by using an inorganic energy. All metabolic reactions in living organisms require organic sources of energy. That means you have to break down something that is made by a living system. Only thing that the plants do is they transform, they have a mechanism to transform the energy of sunlight to the energy of ATP. And that energy is used for every other process. Alright? It is still ATP that is being used. It is still the high energy compounds that is being used even during the Calvin cycle. Alright? So, we will look at this. It is mentioned here, glucose. Glucose entering glycolysis. What will it be transformed into? It's a pathway. Glucose becomes something. What is that something that glucose becomes? Pyruvic acid. Pyruvic acid. Pyruvic glucose is how many carbon compound? 6 carbon. Pyruvic acid is 3 carbon. What enters Krebs cycle? What enters Krebs cycle? Does pyruvate enter Krebs cycle directly? What enters Krebs cycle? Acetyl-CoA. Acetyl-CoA is what enters Krebs cycle. How many carbons are present in acetyl-CoA? Acetyl, acetic acid, how many carbon? Acetone, how many carbon? 2 carbon. Pyruvate is 3 carbon, acetyl-CoA is 2 carbon. So in this process, before it enters the Krebs cycle, 1 carbon is removed. Removed as what? Carbon dioxide. So carbon to become carbon dioxide, what do you need? Oxygen. Carbon to become carbon dioxide, you need oxygen. Removal of carbon from a substance, what can we call that process? Decarboxylation. So we are doing decarboxylation of pyruvic acid. What are we using to do that? What are we using to remove that carbon and make it into carbon dioxide? What is essential here? Oxygen is essential. So what is this decarboxylation? Oxidative decarboxylation. Between glycolysis and Krebs cycle, there is oxidative decarboxylation, which is a single reaction. Now suppose there is no oxygen. Can oxidative decarboxylation happen? No. Oxidative decarboxylation cannot happen. That means pyruvic acid cannot be converted into acetyl-CoA. If pyruvic acid cannot be converted into acetyl-CoA, there is no entry point into Krebs cycle. So pyruvate will go into lactate metabolism. Correct? Pyruvate will get converted into lactic acid. So that process is energy consuming. It is not energy producing. You understand? So the net energy produced in aerobic respiration and anaerobic respiration is grossly different, because most of the energy is produced or liberated in the Krebs cycle. Byproduct of glucose breakdown, pyruvic acid cannot enter Krebs cycle if oxygen is not available, because for that to enter Krebs cycle, it has to be stripped of 1 carbon and converted into a 2 carbon acetyl-CoA. Then it enters Krebs cycle, and there it goes through certain process, releasing NADH, FADH2 and all that, which goes into the electron transport chain, producing ATP and goes on like that. Alright. So 4 steps are there. There are 3 pathways and 1 single reaction. Glucose has 6 carbons. Every carbon linked to each other by bonds. Correct? Energy is in the bonds. So what is your purpose? You have to do 2 things. Break down glucose into individual carbon units. So 1 glucose should be converted into what? Physically, chemically? 6 carbon dioxide. 1 glucose should be converted into 6 carbon dioxide. Now when you break each of these bonds, energy is released. You need to have a mechanism to capture that energy in high energy compounds. Push those high energy compounds into ATP production factory, which is in the mitochondria. So in this entire process of converting glucose into carbon dioxide and harnessing the energy of glucose and production of ATP, what are the energy compounds that are produced? At different places you will get ATP directly through substrate level phosphorylation. You get how many ATPs in glycolysis? You get 4 ATPs, 2 are used. So net ATP production is 2 per molecule of glucose. What in glycolysis? NADPH. How many NADPH? 2 NADPH per glucose. So 1 NADPH is equal to 3 ATP. When it goes to electron transport chain, it will give 6 ATPs. Glycolysis can give you 8 ATPs in general. It is not 6, it is not 1:3. It is 1:2.5. But for calculation purpose, for the ease of understanding, we consider 1 NADPH is equal to 3 ATP. Okay. Then what else is the energy compound produced? In Krebs cycle, we have 3 energy compounds produced. GTP is produced. GTP can be utilized like ATP. So you don't have to convert that into ATP again. GTP can be utilized like ATP directly. You get NADPH, how many per glucose molecule? 6. You get 6 NADPH per glucose molecule, you get 2 FADH2 per glucose molecule. Alright, so how many 6 NADPH is per, not per glucose molecule, yeah, 6 and 4, 836, yeah, 4 FADH, 2 FADH2, no? So NADPH, FADH2, GTP. NADPH and FADH2 goes into electron transport chain. Now, look at this. Glucose is entering. What comes out? What can be 2? What is this 1 here? What else enters glycolysis? What is the first step? Glucose becomes glucose 6-phosphate. So where is the phosphate for phosphorylation of glucose coming from? To take it from somebody, no? ATP. So in the first step of glycolysis, ATP is entering along with glucose. So this is formed into pyruvic acid. What is produced? 4. 4 can be considered as ATP because 3 is going into pathway C. Pathway C is electron transport chain. What is going into electron transport chain that was produced in glycolysis? NADPH. Correct? Now here pyruvic acid also enters Krebs cycle by getting converted into acetyl CoA. So 5 here is a representation of acetyl CoA. It should have ideally been 2 becomes 5 and 5 gets into pathway B. That would have been a better representation. I did not correct it because given in the NEET paper. So we will expose the mistakes also, representational errors. Alright. Here pathway B, Krebs cycle, you have carbon dioxide and water, GTP, NADPH. Carbon dioxide, water, GTP, NADPH, FADH2, carbon dioxide. So you have to assume between 6, 7, 8, you have to assume all of this. Carbon dioxide, water, NADPH, FADH2, GTP, all of that. Alright. Because they are asking you what 4, 8 and 12 mean. 4 we know already is ATP. 8, so therefore from the question we deduce that it might be a reference to GTP. We can consider that to be ATP only. And pathway C is producing 2 things, 11 and 12. 11 is water. 11 is water and 12 is ATP again because 4, 8 and 12 is being asked in the question. At the end of electron transport chain, we will have water being produced, protons, electrons that are flowing through the electron transport chain and oxygen combined together to produce water. That is called metabolic water. There is an animal which will not drink water, which will utilize metabolic water. Which is that animal? An animal, a desert animal, which will not drink water. You don't have to give it water. It will utilize metabolic water. This is metabolic water produced during oxidative phosphorylation. What is the animal? Correct. Kangaroo rat. Kangaroo rat does not need water. It can utilize this metabolic water for its metabolic needs. Alright. So this we understood. Is it clear? The entire purpose of all of this is to break down glucose completely into carbon dioxide, harness as much energy as possible to make what? ATPs. Alright? To make ATPs. That's all. There are a lot of things that we have to see in these processes. That's all. This is the entire process. What we have to understand is enzymes, what kind of enzymes are acting. There are kinases, there are phosphorylases, there are dehydrogenase. We have to understand the enzyme categories. Enzyme categories can be understood using these concepts. Then we will have to understand which reactions are irreversible reactions. Now these irreversible reactions will act as the rate defining reactions. And from there, the irreversible reactions the substrates or the substrates of the irreversible reactions or the products of the irreversible reactions enter different pathways. They enter different pathways, lipid synthesis pathway. So all of that wherever required we will be able to understand. For each reaction we need to see whether that is an energy producing reaction or an energy consuming reaction. We need to know where ATPs are produced, where energy transportation is happening, how the electron transport happens, the redox reactions, which is getting reduced, which is getting oxidized, different complexes of the electron transport chain. And finally, by the end of this discussion, we should be able to understand how is glucose broken down into carbon dioxide completely, how every bit of energy that glucose has is converted into ATP. Am I right in saying that? Every bit of energy that glucose has gets converted into ATP? Why not? A major portion of that is lost in the form of heat. So when we are discussing only ideal situations, chemical situations. Actual metabolic situations will differ based on the conditions in the body. We need not go into the detailed elaboration of that. We need to know these things that we are knowing. Alright. This entire process will just open up your mind. It will open up your mind for understanding the entire chapter on photosynthesis. Alright. So with only 4 questions, 3 hours, we have been able to deal with all the major concepts related to plant physiology. By the end of the week, before I leave, you should be able to answer questions related to plant physiology and human biology very well. Alright. I will not rush in discussing. I have no target of completing the discussion of every question in the post-test analysis. My target is to take you through elaborate discussions of as many questions as we can, which will further train you to do it yourselves. Alright. So you should continue doing the post-test analysis in a sequence that you are following. Don't jump from paper to paper. Complete one, go to the next one. It's okay to have a lag, but just remember this. 3 things I told you for post-test analysis. Number 1, PDF, purpose, design, function. The second thing that you have to look at is what are the terminologies that are there. You have to understand the terms and get to know about it. And the third one is what? Ask more questions. Don't just limit yourself to PDF. Ask more questions to get diverse responses from your own exploration. Alright. So let's keep doing that. We'll break for the day.